Hello, and welcome back. We're going to continue talking about genetic traits. All right, we left off talking about dominant and recessive. Okay, with that being, the dominant trait, you only need one copy of that allele or that gene in order to see that particular trait. Okay. Something else I want to kind of define here is what a phenotype is versus a genotype. Okay? Because this is going to be important with genetics. All right? When we're talking about phenotype, we're talking about what you see associated with the particular trait. Okay? So a phenotype can be eye color, brown eyes versus blue eyes. A phenotype can be flower color, purple versus white. It's something that you physically see. Okay? The genotype is going to be what you've got at the DNA level. Okay? So we're talking really with the genotype, the particular sequence that you've got for that particular gene, but the way we represent genotype, okay, genetically, we're going to represent it with letters. All right? And those letters can represent the dominant allele or they can represent the recessive allele. Okay? For dominant alleles, we're going to use the uppercase letter. The recessive allele is going to be lowercase. Okay? And in defining the alleles, there's a little bit of variation in this. What I like to use when I define alleles in genetic crosses are, I try to use letters that it's easy to distinguish upper and lowercase, okay? So with our traits here, we've got purple and we've got white, okay? P's are hard to distinguish whether you're putting a capital or a lowercase unless it's next to the other. White is kind of the same way, okay? So, you can pick any letter you want, okay? We can go with, in this case, we can use an F since we've got flowers. We can use a capital F and a lowercase f. Those are easy to distinguish. I think in the PowerPoints, I use a capital A versus a lowercase a. A's are also good letters. It doesn't matter what letter you use as long as you define it, okay? So if we say F is going to be our purple flower, lowercase f is going to represent white, all right? So if you notice, we kind of mix when we're talking about genotype and we're talking about phenotype, okay? The genotype, we're talking the genetic level, okay? In this case, this particular flower, the purple color, is going to be the dominant version of the allele, okay? So that dominant version, you can kind of think of it in a way as being a little bit stronger. It can mask the presence of a white allele, okay? So if we want to define this genetically then, okay, we could say our alleles are capital F, lowercase f, okay? And that combination, when the two cases don't match, we refer to that as being heterozygous, okay? Hetero meaning different, okay? We also can have some other combinations. We could have two capital letter F's, all right? This would then be considered homozygous dominant. We could have 2 lowercase f's. This would also be considered homozygous. But now because we've got 2 lowercase alleles, lowercase represents recessive, we would call that homozygous recessive, okay, as far as our terminology, with the homo prefix just meaning same, okay? So this is going to be important because we're going to start following some of these traits, and the way we're going to follow these traits is using what's known as a Punnett Square. And this is just a way for us to kind of keep track of the alleles and to predict, I predict I've got a squeaky marker again, to predict what the offspring will be, okay? So we're going to start this Punnett Square and we're going to define and use this as a monohybrid cross, okay? With a monohybrid, mono meaning one, okay, you are tracking one trait, okay? In this case, the trait that we're tracking is going to be flower color, all right? So what we're going to do, we've got up here our heterozygous example. We're going to use that as one of the parents, okay? So our parental generation is going to be heterozygous, and we're going to cross it to another heterozygous plant, and we're going to look at the outcome from that, okay? So our parental generation, we're going to set this up, okay, next to our boxes. Now, your lab instructor may have a particular way. They may want females on the vertical here, males horizontal. To me, it doesn't matter, because really what you're doing is you're tracking what's going to happen with the offspring, all right? But if your particular instructor is fussy about that, keep that in mind, okay? If you work through the problem, you're going to get the same result whether you put mom here or mom here, as long as you have the alleles assigned correctly, okay? And the monohybrid cross is going to be our simplest one, all right? So then what we do to fill in our Punnett Square, we're going to take our plant that's heterozygous for our flower colors, and we're going to see what the offspring potentially could be genetically, okay? And the way we do that is we look at the box and we look at what letters match up with that particular box. So this first box here, we've got a capital F, okay? So that's one genetic combination that can come out of this. If we go across, we still would put in the capital F from over here and our lowercase f from up here, all right? This one, same thing. We follow it down. We've got a capital F, so we could put a capital F in this box, lowercase f, lowercase f. This one, lowercase f, okay? Now with genetics, something that you do need to do and follow for it to be proper genetic nomenclature, okay? The dominant allele is always put first, okay? So if you did this and you looked at this particular box here and you put little f and then big F, genetically that's not correct, okay, as far as naming. You always want to put the dominant allele first, okay? Now what we have inside this box is our F1 generation. Oh goodness, we're squeaky again, I apologize. So the offspring genetically can be capital F, capital F, capital F lowercase f, capital F lowercase f, or 2 lowercase f's, all right? And those genotypes now will tell you something about phenotype, okay? Capital F lowercase f are going to have purple flowers because those are the dominant traits. Lowercase f, because we have 2 recessive alleles, because we need 2 recessive alleles to see that trait, our flower colors would be white, okay? And what you're going to notice when you set up crosses like this is you're going to notice some ratios come out consistently when you have certain genotypes associated with the parent, okay? With this one, I set up 2 heterozygous parents, and if you notice with our phenotype, we've got a 3-to-1 ratio, meaning we've got 3 offspring that are going to have purple flowers, 1 offspring that's going to have white, okay? Now out of this we can also, I'll put this up here, look at the genotype ratio, okay? Genotype ratio is going to be a little bit different. With the genotype, we've got F, capital F, lowercase f, or lowercase f. And let me make those colons, okay? Now, when we look at that, capital F, we've got 1. Capital F, lowercase f, we've got 2. Lowercase f, we've got 1, okay? So when you cross 2 parents that are both heterozygous, you will always see the genotype ratio of 1 to 2 to 1, okay? And a phenotype ratio of 3 to 1, okay? That'll be consistent if the parental generation, both of them are heterozygous. Now, if we change the parental generation, we're going to change some of what our ratios are. I'm going to, in the next video, kind of show you some of those differences with changing the parental generation, okay? So hopefully that made sense. If you have any questions, please let me know.